All right. Welcome back, everyone. Thank you for joining us for our final talk. And this is a keynote talk of the conference. And it's my pleasure to introduce my friend and colleague, Dr. Eduardo Eizirik from the Pontifical Catholic University of Rio Grande do Sul in Porto Alegre, Brazil. And Eduardo is an internationally renowned scientist who's worked on molecular ecology, mammalian evolution, coat coloration evolution in carnivore species, and he's one of the foremost leading experts in carnivores and mammals, and very famous in South America for sure. And he always gives great talks. And you know, for some reason, you know, we've had a lot of carnivore talks this morning: ferrets, brown bears, wolves, and now we're going to listen and hear about carnivores in South America. So welcome, Eduardo, and thanks for coming and giving this talk.
Thank you, Carl. Thanks, Klaus. Thanks a lot. Is this mic working? Can you hear me, people online? So first of all, thank you, Klaus, and I'd like to thank the organizers as a whole for inviting me to give this talk. It's a pleasure and an honor, and I've enjoyed this conference very much. I mean, it's one of the best conferences I've been to in a long time. I thought the talks were excellent, so I hope you like the examples that I brought here to show you. So as Klaus mentioned, I think now it's on. I'm a professor at this university in southern Brazil. I'm also a member of an NGO working on carnivore conservation and of this National Institutes of Science and Technology on Ecology, Conservation, and Evolution. So I've been working on evolutionary biology and conservation biology for about 30 years, mostly focusing on carnivores. So just to place carnivores into context, and of course, as Klaus just said, you probably don't need that. But I'll start by just showing where they are in the tree of life. This is a mammal tree we published about 11 years ago. And we've been continuing to work on the mammalian phylogeny, but the conservation-oriented work that I do is mostly pertaining to carnivores. So who are they? They're a monophyletic group. It's an order within the mammalia. So I just brought you some examples, and you've seen several during the conference as well. As you know, they have quite a bit of functional diversity. That's the largest span of body size among mammals. You have aquatic species, and terrestrial species, arboreal species, burrowing species. So they're very diverse, and from an evolutionary perspective, a very interesting system to study diversification, speciation, and other interesting processes. But also, it's a group of considerable conservation concern. Right? You have many problems related to habitat loss, human-wildlife conflict, poaching, and many others. So they are also a very relevant topic in the context of conservation biology. In my group there at PUC, this university that I work on, I've been tackling several of these problems. I'm not going to go into each of them, but we've used a variety of approaches to study different carnivorous species over the last 25 years or so, merging ecological, genetic, genomic approaches and different analytical strategies. And I brought you here some examples that I'm going to go into. Starting from a phylogenetic framework that we can look into the different groups of carnivores that we have been studying. This is a study we did in 2010. This, I think, was the first paper that Klaus and I co-authored. So it's been 12 years already. This is a phylogeny of the carnivora, showing that it has 16 different families. This is a phylogeny that dates the origin of the different families and that shows the relationships. An interesting feature of carnivores from a genomic perspective is that it has two very well-known companion animals representing its two main branches. So the filiform carnivores, it's a suborder, and the caniform carnivores. This is, by the way, my dog and my cat. I just put both as examples of the two main branches. They've been separated for about 60 million years. So if you have a dog and a cat at home, you have things that have diverged over 60 million years ago in your house, which is kind of cool. And this also translates to good genomic resources. So we can leverage on the genomic resources that are made available by these companion animals for a lot of our research projects. And this is actually complemented these days by additional genomic resources that have become available recently. For example, a Canadian lynx VGP genome that became available recently that's very high quality. And so not only do we have a domestic cat, good quality genome, but we also have a Canadian lynx genome that we can use as references from several of our comparative projects. Also here at the Sanger Center, they sequenced a Eurasian otter, and that has also recently become available. It's a representative of another family, the Mustelidae, that we just heard about regarding the black-footed ferret. So we have a good quality otter genome. And there are several other good quality genomes for many of the carnivorous species that we have been studying, and this will tend to increase in the future because these are high profile and very studied species. Most of my work has been focusing on the carnivore families that occur in the Neotropics. So the Neotropics are these regions here, comprising South and Mesoamerica and the Caribbean. These families highlighted in green here are the ones that have invaded and colonized and diversified in the Neotropics. And we have been studying their evolution for a while. So this is a dated comparative tree of the different terrestrial families of carnivores and when they diversified. This is a time scale here, and the gray bar is the timing of the Great American Biotic Interchange, which is a time when the Panamanian Isthmus was formed, and there was this large faunal interchange between North and South America, and many of these carnivore groups invaded South America coming from the north and radiated there. So for example, we have cases here within the cat family and the dog family of very recent radiations. So these are genera that have radiated in South America after the invasion by a single ancestor. So this provides us with interesting opportunities to investigate species delimitation, divergence in adaptive traits, and several other evolutionary topics. Moreover, in the case of the, did it show? Yeah, there's another highlight here on top of it. There's a recent result that we got, I'm going to show it to you later, indicating that not only did this genus probably radiate in South America, but the whole canid radiation occurred in South America. This whole subclade within the canid, the South American clade, radiated within South America after a single ancestor invaded the region. So you can look at it at different temporal scales, you can look at it at different spatial scales. There are many evolutionary problems that we have been studying over the years, but here I'm going to mostly focus on this final aspect here. Several of these populations and some whole species are threatened by a variety of problems: habitat loss, fragmentation, human-wildlife conflict, and there are many unknown aspects about them. Some of them don't even have their taxonomy well resolved. We don't know what the species are. We don't know how different the populations are. So working out the basic evolutionary framework upon which we then work in conservation-oriented projects is a priority. So we've been doing that as well, and increasingly using genomic approaches. The first genome that we sequenced was the Jaguar genome. This was the first genome project that took place in Brazil. We started it back in 2011. We sequenced the genome in 2014 and published it in 2017. It was a consortium of Brazilian institutions that worked together to generate the genome and annotate the genome, assemble it. And then several international partners from these other countries worked with data analyses and helped us understand the genome and characterize it and then study it in more detail. This is the publication that came out of it. It was a very close collaboration with Bill Murphy from Texas A&M and his group, especially Gang Lee, who's the co-first author with Enrique. Enrique, who now works with Klaus, is here. This was his PhD project. And so he and Gang Lee worked closely, and several other collaborators are here. This was an interesting project, not only in terms of capacity building in Brazil, because it was a major challenge to do a whole genome sequence there at the time, but also allowed us to perform a variety of analyses. We included a leopard sequence that we produced for this project as well, and we had sequences from all the big cats. It was the first time that we had whole genome sequences for the whole clade of big cats, the Panthera genome. This is another cat that I had, so it's another of my cats that I used for the figure as an outgroup. I'm not going to go into the phylogenomics very much. I'm just going to mention briefly a couple of results because it has to do with things that we discussed here at the conference. This is the tree that emerges from the genome. This is the most prevalent topology in the genome, the relationship among the big cats. But an interesting finding was this one here at the bottom. This is the whole genome-wide alignment of 2.4 gigabases, and these are the domestic cat chromosomes, so it's aligned onto the domestic cat chromosomes. At the time, the only chromosomal scale genome we had for the Felidae was the domestic cat genome. And these are 500 kb windows, slid 100 kb at a time. So basically, it's a sliding window analysis of the topology. And in blue, you have this topology. That just shows that this topology is prevalent across the genome, it's up to 95% of the genome, depending on the size of the windows that you analyze, from 75 to 95%. And in red, there are the other topologies. So there are 105 theoretical topologies in a 5-species rooted tree, and all 105 are present in their genome. So basically, there's a lot of genealogical discordance. That's why I brought this up earlier in the conference. And we observed that the topological discordance is concentrated in the central regions of chromosomes, which is kind of an intriguing result. And we also found that there's a lot of evidence for ancestral admixture in this group. So this is the main topology. Okay, it stopped working. Can I have another? Well, I'll just talk about it. There's nothing pointing, right? Yeah, it's going to come. So the arrows indicate ancestral admixture events, interspecies admixtures. And so you see there's plenty of it across the tree. I'm just going to move to the next one. I don't think I need to point this very much. This is a subsequent study we did with 27 phyloid genomes, exploring the genealogical discordance some more across each of the phyloid genera. This is just showing the results for the Panthera genus. Up on your right, all the red dashed lines indicate ancestral admixture episodes between genera. So this is really beside the point here at this conference where we're focusing more on intraspecific problems. But this just shows you how much genealogical discordance there is and how much of an issue it is for several analyses that we perform, including detecting signatures of selection, including estimating population parameters, because a large fraction of these genomes actually contain admixed segments. They come from other species, not only recently, but also farther in the past, right? So the dashed lines there indicate intergeneric admixture events. And then within genera is the focus, within the genus, is the focus of this main figure here. Maybe it came back by itself. No, it hasn't. I'll just keep guiding you through the figure here. Topology 1 places lion and leopard together. It's our main, it's our most common topology. Topology 2 puts lion and jaguar together. Topology 3 puts jaguar and leopard together. These are the three most common topologies. There, to the right of it, you see those histograms, those bar charts for autosomes and the X chromosome, and you have low recombining and high recombining regions. So we think the proximal versus distal portions of genomes have to do with the recombination landscape. Basically, the areas of low recombination have one phylogenomic pattern, and the areas of high recombination have another phylogenomic pattern. So what you see is that the topology 1 is prevalent in high recombination regions on the X and on the autosomes, but topology 2 is more prevalent in the low recombining regions. And if you look at the other chart there, the other graph at your bottom right, you'll see a very neat relationship between recombination frequency. So these are quantiles of recombination across autosomes on the left and the X chromosome on the right. And you see that topology 1 increases as recombination increases, and then topology 2 decreases. So basically, there's a very clear relationship not only on the X, but also on the autosomes. Here in this main one here on the left, you see the recombination map for the domestic cat. Thank you, Klaus. Thanks a lot. All right. So here is just the X, and we have that for the other chromosomes as well, where you see recombination frequency and topological frequency. In blue is topology 1, and here are other topologies, especially topology 2. So basically it just shows that the other topologies, especially 2 and then 3, are enriched in low recombining regions of the genome. And that's just kind of an add-on to this talk, because I thought it was interesting for you to have a feel for how recombination-aware phylogenomics is evolving, and it has an impact on other inference problems that we have, including intraspecific inference problems. So as a consequence of these genomic studies, we have now accumulated almost 400 genomes sequenced for Neotropical carnivores. We've sequenced by now all the cats in the Neotropics, including the jaguar. I'm going to spend some time on the jaguar later on in the talk. All the canids, I'm going to mention that. The otters, all the world's otters, including the 4 Neotropical species, and a lot of smaller cats. This is going to be the last example. I'm going to talk about all 4 examples. Thank you. But Klaus gave me one, so yeah, we're good. Okay. And I'm going to focus on these 4 papers that came out this year. So those are the papers that describe some of these findings, plus some unpublished data that I'm going to tell you about as well. One example of a study that came out this year was led by Jorge Ramirez. He's a Peruvian scientist, was doing a postdoc in Brazil in Pedro Galetti's group and collaborated with my group. He's back in Peru, he's a professor now. In this project, we sequenced the genomes of a margay, which is a very arboreal, it's the most arboreally adapted wildcat in the Neotropics, and an ocelot. We already had an ocelot genome from a previous study, but now we have a second one from the wild. The previous one was a captive individual. We've moved away from captive individuals as much as we can because we have very limited inferential power with captive animals. So we've been focusing our genomic sequencing as much as we can on wild animals. I'm just going to go through some of the results. If you look at heterozygosity across the genome, these are the ones from this genus that we have sequenced and that we had sequenced before. And these are other cats from other genera that had been sequenced before and that are the most variable species, the most variable individuals that had been published so far. So what we're showing is that the ocelot, this species here from the Neotropics, this is the range of the ocelot, is the most diverse cat so far. So it is the cat with the highest genome-wide heterozygosity. It has to do probably, so this is a PSMC plot for the ocelot, and we have the two individuals, the captive individual and the wild individual, so they mirror each other fairly well in this case. And you see the PSMC plots for the other Leopardus genus species, and you see ocelots seem to have had larger effective sizes, and probably it has to do with higher densities. There are ecological data sets indicating that it has higher densities in most areas. It's very generalist in its ecology. It has a broad range. It doesn't have a lot of structure, at least south of the Amazon, which is where this sample comes from. It's not very structured. So there are interesting features that, of course, will follow up with population level sampling eventually. A different pattern emerges. Now I'm just going through some of the other cats without the ocelot, just for the scale is different now. So this is the northeastern tigrina. It's a small cat sampled from here. And we do think it's a species complex, as I had mentioned earlier to some of you. So we don't think that this necessarily represents the whole orange here. It's mostly this northeastern Brazilian population. But we see a fairly sharp decline and very low heterozygosity in this species. And we're now sequencing multiple individuals to understand its demographic history in more detail. An interesting pattern emerged here. This is the arboreal species, the forest-dwelling margay, and this is a pampas cat, which is a grassland species. And it's the first time that we see this kind of PSMC pattern, these alternating demographic fluctuations. One is a grassland species that probably expanded during times of glacial maxima because grasslands expanded in South America and forests dwindled, and vice versa. So they seem to have these alternating demographic histories in South America. And this is also a species complex that we're trying to sort out, and this is just one of the populations that represent the species complex. Going into the next example, I'm just picking some features from some of the studies and trying to focus more on the demographic aspects and less on the adaptive aspects. All of these studies have had parts of them that were focused on detecting signatures of adaptive evolution, and I'm not going to include those in the talk today so that we have more time to discuss conservation-oriented questions. This one came out this year as well. It was led by Daniel Chavez. It was a collaboration with Klaus and also with Bob Wayne. Daniel was Bob's PhD student. He's from Ecuador. He's back in Ecuador now. And the idea for this project started back in 2011, at the same time that we came up with the idea for the Jaguar genome project. The initial idea was to sequence the genomes of the maned wolf and the bush dog, which are very disparate species. These are sister species that have diverged a few million years ago. That's one of the things we dated here in this study. But they have become extremely different from a morphological standpoint. One has long legs, the other has short legs. This is solitary, this is social, this is hypercarnivorous, and this eats fruit a lot. About 50% of its diet in some areas is fruit-based. So they have become quite different, and our initial idea was more of an evolutionary focus project on these two species, basically understanding the genomic underpinnings of their morphological, ecological, and behavioral differences. But it ended up growing and became a genome-wide assessment of the evolution of all South American canids. So this is the whole radiation of South American canids. It has been very hard to resolve phylogenetically because it's a rapid radiation. There's lots of ILS. There's interspecific hybridization as well. These are the ranges of these species. They are color-coded, so you can see based on their colors where the ranges are. We split them into 3 different maps. This is a biogeographic reconstruction, east of the Andes, west of the Andes, and central, or both sides. And you can see this biogeographic reconstruction of when they occurred. When is in the next slide, but where they occurred here. We can see that most of the early divergences seem to have occurred east of the Andes. So we reconstruct the history of this radiation occurring after entrance of a single ancestor into South America and then radiating east of the Andes, then circling the Andes here in its southern area, and then some of the latest splits occurred west of the Andes. So the western clade is nested within the eastern clade, and we can infer a sequence of split events that led to this radiation in about 3 million years. This is the analysis that Hélène Gronau ran, and gfox is one of the main collaborators in this study. So here we see the same topology, but the width of the branches is proportional to the effective size at the ancestral nodes. So you can see the timing of the South American radiation, about 3.5, which fits well with the closure of the Panamanian Isthmus. So this lineage entered South America and then radiated. You have some small effective size, and some others are larger effective sizes here. You have population expansions that you can infer based on the diversity that you reconstruct at these ancestral nodes. You have some inference here of hybridization, of interspecies hybridization. And here in this group here, this fox-like group, you have lots of hybridization. This blue bar here indicates a lot of ancestral admixture. So not only do you have a lot of ILS, but also a lot of interspecies admixture. So this is an interesting model upon which we can now understand the demography of these species in more detail. This part of the figure is an estimate of interspecies admixture, pairwise. So the source species is here and the target species is here. You see, for example, a lot of inferred hybridization between the pampas fox and the South American gray fox, two species that are morphologically quite similar. There has been a constant taxonomic debate on whether they are the same species or two different species. And that result here indicates that they have indeed hybridized a lot in the past. And so our next steps are to do more individuals of them and to study them on a population basis across a transect in southern South America. Another side note here is that there is quite a bit of evidence of hybridization between the hoary fox, which is the only Brazilian endemic fox, which is this one here. It's around here. It's endemic to the Brazilian Cerrado. It's a savanna-like environment in Brazil. So there's plenty of evidence for hybridization between the hoary fox, which is the one that diverged earliest in this clade, with the pampas fox. So the pampas fox has hybridized with at least two different species in the past. One study that I did not bring here to the talk, but it has to do with this observation, is that the individuals we use here were the non-admixed individuals, because we have discovered a hybrid zone, an anthropogenic hybrid zone between the same two species around here. So in this region here, these two species are hybridizing now. These are grassland species, these are open habitat species, and they have been separated by hundreds of kilometers of Atlantic forest that humans have now depleted. So now they're expanding in both directions and hybridizing. So they're currently hybridizing due to human-induced habitat alteration, but the pure individuals that we did not pick up as hybrids also have more ancient signatures of hybridization, indicating that this has probably been a cyclical dynamics of expansion and contraction. They have hybridized in the past. Now they weren't hybridizing. Now humans are inducing hybridization again. It's just a side note on this result. We have estimated genomic diversity. So this is heterozygosity plot. So heterozygocytes per kb. You have species with high diversity, especially the smaller foxes. You have species with low diversity, such as the maned wolf, the bush dog, and some others with low overall levels of diversity. Here we have a focus on Darwin's fox, which is an endemic species here to this coastal area of Chile. This is a picture of it. It's a threatened species. You have clearly runs of homozygosity across its genome. Here's another figure where you have the mean heterozygosity per kb for the species. And here's the sum length of the rows, the runs of homozygosity. You see the two individuals with the most runs of homozygosity are two individuals from an island and from the continent belonging to this species, the same endemic species that we see here. PSMC plots for the whole group. I'm not going to go into all the different subgroups in there. I'm just going to show, for example, here you have multiple individuals, wild-caught individuals of the maned wolf versus the bush dog, which are that pair that we wanted to study. The bush dog is more forest associated and the maned wolf is more grassland or cerrado associated. And you see this diverging trend here that seems to coincide with the glacial maximum, where the open habitat species remain stable and the forest dwelling species declined. My main focus here will be again on the Darwin's fox from this coastal area of Chile. You see, if you take a look at the scale, it has very low effective size and very sharp decline trends recently. So it's a species of conservation concern. This is now the basis of discussions on population level analysis targeting the species. Of course, there were already conversations about the conservation strategies on behalf of the species, but now we add a genomic component to this and show that it has less diversity, more homozygosity. The two populations are different in their diversity patterns, and so that's an important aspect for us to consider. We're talking to Chilean colleagues about this as we move forward. The next study that I wanted to highlight was also a close collaboration with Klaus. Klaus and I co-advised Vera. Vera was a PhD student at my university. And this is her main PhD paper. It came out as the cover of Current Biology a few months ago, so we're happy with the cover. This is a Chilean otter species, Lontra provocax. I'm going to talk about this species in particular in a bit. We used 24 genomes here. 14 of them were novel, and 2 were generated from museum specimens, which was important for us to be able to sample all 13 currently recognized otter species from across the world. And 2 of them were from museums. This one from the Congo Basin, and one of them from Sumatra. So it was nice that we could use museum materials to generate whole genome sequences, of course, with a lower coverage than the fresh samples, but it worked out well. This was a collaboration with Tom Gilbert's lab. This is a phylogeny we reconstructed from this whole clade. We were able to robustly resolve the phylogeny, date the origin of the different subclades, make taxonomic propositions. And I'm just going to highlight this pair here, which I think is one of the conservation implications of this study. This is that Congo Basin species that has been very poorly studied. Up to now, there were only a few mitochondrial segments, a couple of mitochondrial segments generated from the species, and there was discussion over whether it's a separate species or not. So we had 2 individuals each of Conjicus and its sister species, the broadly distributed Capensis, and we could show that they're reciprocally monophyletic at the genome level. Their divergence is over 400,000 years ago, which is on a par with this other pair of sister species from South America. So we have argued that it is a distinct species that warrants conservation concern in its own merit. So that's one of the conservation implications there. We measured genomic diversity. So here's heterozygosity, mean across the genome. This is the red list category of the different species. So we're comparing levels of genomic diversity and red list categorization. This is an analysis of runs of homozygosity, looking at different sizes of runs of homozygosity. So I'm going to highlight particularly here these 2 southern South American species, Lontra provocax, which is the one that's on the cover of the study, but also Lontra felina. It's a marine otter. It's a convergent species relative to the sea otter of the North Pacific. So it's a marine species that also has low diversity. Both of them are considered endangered. Both of them have low diversity, and both of them have quite a bit of their genome covered by runs of homozygosity. And the one with the most dramatic examples of runs of homozygosity is Lontra provocax. If you look at SNV density across the genome, this is a broadly distributed Neotropical species. This is a heat map with the warmest colors indicating more diversity across the genome. Each bar is a chromosome, and you see the distribution of SNVs across the genome. And provocax, this species here, has very low diversity across the genome. These are PSMC plots for the different species. They have different scales, so you see some species with much larger effective sizes, others with smaller. Many of them have this trend of decrease, increase, and decrease again. They're not exactly at the same time, but some of them do seem to be at the same time. They seem to correlate with glacial cycles, and it's interesting to compare them across regions and within the same region, so we made some of those comparisons in the paper. Again, I'm going to highlight provocax. This is the range of the species. It's another species from southern South America, mostly Chile, but also Argentina. Look at the scale here. See, the scale is much smaller. So it's a species with very low effective size, very recent decline, very recent coalescence. So quite remarkably different from the other species of otters across the family. So looking at these patterns and also considering the fox example, this brought to mind another study that a PhD student from Belgium is pursuing in my lab in collaboration with Hannes Svardal's lab at Antwerp. This is Jonas Lescroart. He's now spending some time in Beth's lab in the US. This is one of his PhD papers. He's looking at that genus of small cats that I have mentioned before, genus Leopardus. One of his studies includes complete genomes from all the species, 17 different individuals. And this is one of the species that we have sequenced recently. It's a guinea. It's a Chilean endemic species with some populations in Argentina. Here on the west of the Andes, this is its range. Has very low diversity as well compared to the others. The ocelots I have shown before, we now have a third individual and they continue to be very diverse. There is variation. We now have an Andean mountain cat, which is a very rare species from the high Andes, also has low diversity. But the comparison here that's interesting is the codcod or guinea with low diversity and its range. And it also has a similar pattern of decline in its PSMC plot with low effective sizes for a long time. And I thought it was interesting to put the three together. So we have the otter from this region, you have the fox from this region, and this is unpublished, but we also have the cat from this region. So we have three different species from three different families, all three occurring in that region of southern South America, and all of them showing similar patterns of low effective size, recent coalescence in their PSMC plots, declines in their PSMC plots, low heterozygosity. So that indicates a common biogeographic pattern, probably driven by glaciations. So a lot of those regions were covered by ice sheets. So there's a lot of study of glaciation effects in the northern hemisphere, but not a whole lot in the southern hemisphere. So this is some of the first genomic evidence of likely effects of glaciation cycles in southern South America, leading to concordant patterns of genetic diversity in different species occurring in the same region. So this opens up several different avenues for us to investigate not only evolutionary, but also conservation-oriented issues regarding their low diversity levels. A quick show of another study, also by Vera, also in collaboration with Klaus, on otters, just because I thought this was a conservation-related study. We did this study on the 13 currently recognized species, and now that it's done and it's published, we can go on to publish this one, which shows another species. So now there's 14, actually. Vera used UCEs in this case, ultra-conserved elements, not whole genomes. So this analysis uses about 30,000 SNPs across the genome to look at Lontra longicaulis. So this is the broadly distributed species that has the most diversity, the one that I showed in the plot with the warmest heat map of diversity across the genome. Has lots of diversity. It occurs throughout Mesoamerica all the way to here, northern Argentina. It does show intraspecific variation in the rhinarium shape, which is this nose pad. And this in other otters is indicative of species-level differences. But this was the only otter species with intraspecific differentiation, which has suggested that they may not be a single species. There were some mitochondrial DNA evidence, some morphological evidence, but it was still quite incipient. So here we sampled the individual where these dots are. So we collaborated with these Mexican scientists who are listed here, and included Mexican and a Colombian sample as well. So what we find, this is an admixture plot assuming 2 populations, and this assumes 3 populations, was that the subspecies that had been proposed, these are the 3 subspecies, and these are the ranges of the subspecies in pink, green, and blue. They do correspond to genetically distinct groups, especially the Trans-Andean one, so to the west of the Andes, the pink one corresponds to a very distinct group. And if you go into another level of population structure, you also see the Amazonian subspecies as distinct with some admixture with the eastern population. The PCA plot is very dramatic. You see the western population is very distinct, and the 2 eastern populations are still distinct on PC2. If you look at a tree reconstructed from these SNPs, it's quite remarkable because not only are they very distinct, but they're not monophyletic. Basically, these are the other 2 South American species. This is the one with the lowest diversity that I mentioned. This is the marine one that I also mentioned. And they're actually contained in the diversity of Lontra longicaulis. So what you actually see is that the Trans-Andean group diverges first. So this divergence west of the Andes, and then the Cis-Andean group, the eastern group, separates them then into the southern South American one and the eastern one, including the Amazon. So overall, these results suggest that this Trans-Andean population should be recognized as a separate species, which we call Lontra nectans. This is the available name for it. It's now used as a subspecies, so it's just a matter of raising its status to species level. And of course, this has important conservation management implications because IUCN, for example, instead of assessing this whole thing or this whole thing as one species, will start assessing this as a separate species and this as a separate species. And we've done this in the past with other carnivores, and it has had implications in terms of IUCN categorization and management strategies across the species range. So because of its conservation implications, I thought this was an interesting example to show. I'm not so good with time, but I'm going to speed up a little bit and talk about jaguar conservation genomics. This is a species that we have been working on for a long time, doing genetics with traditional markers. And so this is our first study that came out in 2001 on the genetic diversity, population history, phylogeography of jaguars. This is the original range of jaguars in light gray. And this was a simplified and optimistic view of current range of jaguars back in 2001. It's much worse than that, much more fragmented. But that's what we had at the time when we published that study. These are the sampled individuals. Here are some features of the jaguars. It's the largest felid in the Americas. It has been extirpated from over half of its range, has severe loss and fragmentation of its habitat, persecution, poaching, wildlife trade, a lot of problems in terms of its survival. Some strongholds remain, especially in the Amazon here, and in the Pantanal here, and to a lesser extent in the Yucatan Peninsula, but many populations are in dire straits. And this is one of the main findings of this study. There's genetic evidence for historical range-wide connectivity. We've been working on this with a variety of approaches and showing how bad the situation is, especially for the Atlantic forest, which is this area here. I'm going to show maps of the Atlantic forest soon. It was a very large biome with over 2 million square kilometers on the eastern side of South America, in eastern Brazil, but it's been very strongly denuded. Less than 10% of it remains, and it's all severely fragmented. We've been arguing that this is a critical problem for jaguars in the Atlantic forest, and that soon the Atlantic forest, this is another paper we published later on, will lose its top predator. So we have about 250 individuals spread into very small populations of 10, 15, 20 across this large area. This is the gray areas where the Atlantic forest used to be. This is Brazil, Paraguay, northern Argentina. This is where jaguars still can occur in terms of habitat suitability, and the darker ones are where jaguars still are. So you have these small isolated populations with hundreds or over 1,000 kilometers of people in between. So very altered habitat that's impermeable to them, and that we have both genetic and ecological studies addressing that. For example, our first study that was done in 2010 looked at these inner Atlantic forest populations, and you see the structure plot. Each group here is a different fragment, and we've shown that they have, this is microsatellite still, we've shown that they have undergone drift-induced differentiation very quickly in like 40 years. They have become very differentiated due to small effective sizes. We've done that with another isolated population, which is this one in green here. You see it's very different, low diversity, low effective size. Our overall inference of historical connectivity remains. It holds up across our studies. We've done about 10 studies so far on different populations. And the overall conclusion is that historically jaguars were very connected. They were very connected across these biomes. And recently, these isolated populations have drifted away from each other and have reduced diversity. To back this up, we've been continuing to study larger populations such as the Amazon. This is a plot that shows differentiation and admixture. Here you can actually retrieve an original Atlantic forest similarity, genetic similarity, including that isolated population here, so some ancestral alleles remain. Here are the very fragmented populations that are quite different, the Pantanal population, and the Amazon, even though it spans about 3,000 kilometers in this sampling transect, behaves as a single pannictic population. So this has moved on from microsatellites to whole exome sequences, GBS, and other approaches. This was a study we did with whole exome sequences with about 30,000 SNPs, just showing that the different biomes are quite similar. I mean, there's some structure, but it seems to be mostly isolation by distance, not a lot of deep structuring there. This was a study that I thought was worth mentioning here, also by Gustavo Lorenzana, who did this microsatellite study here as part of his PhD. He's a Mexican biologist that came to work on jaguars. This was a comparison with red-seq data and whole exome sequence data, focusing on the flank. So not using the exonic regions, but the flanking regions, just to see if we had concordant patterns using the same individuals. This is something I had always wondered about, whether if we use the same individuals, what kind of concordance we would get with these different reduced representation approaches. And the concordance is fairly good, depending on how you parameterize your filtering. But the main take-home message here, these are FST values between the different biomes. The larger populations, such as Amazon and Pantanal, don't have a lot of differentiation. It's largely related to distance. And the ones with the lowest effective size, the Atlantic forest and Caatinga, they have higher differentiation, which indicates, again, drift-induced differentiation very recently. We moved on to whole genomes. This is Gustavo's third PhD paper. So these are now whole genome sequenced across the range. This was a zoonomy individual sequenced in Arizona, sample provided by Ollie Ryder. This is a Guatemalan individual, and all the others are from the different Brazilian biomes that I've been talking about. These are the different biomes here. So these are 13 genomes that we started studying. We do a PSMC plot. You see this very concordant trajectory, which again supports historical connectivity across the range. The interesting outliers are these two, which are the ones from the US and Guatemala, so the ones on the northern end of the range. We interpret that sharper decline here as evidence for successional bottlenecks. So they have successively colonized the northern tip of its range, and then this has led to lower effective sizes here. So we see this as an effect of colonization towards the periphery of the range. We looked at runs of homozygosity. You see a trend where the Arizona and Guatemalan ones have more runs of homozygosity, but the ones I'm going to highlight are the ones from the fragmented Atlantic forest populations. You have an enrichment for long runs of homozygosity, indicating recent inbreeding. Speeding up a little bit more. This is a newer study led by Caroline Sartor, who's now at Oxford here doing a postdoc, also by another graduate student in my group, Gabriele, and Enrique has helped in this project as well. So these are now 48 genomes. If you take out the Guatemalan and US ones, we have 46 from different points in Brazil. So we're trying to sample the different biomes more densely. And I'm just going to show you some unpublished results on that. This is a PCA plot where you see the different biomes. In most cases, they are not that different. So again, supporting connectivity. But you see these outliers. And the outliers do correspond to the small isolated populations. So these three individuals are from here. This is a coastal Brazilian population that we had not sampled before. You see very strong differentiation there. You see this other set of populations here. That's another fragment. Two fragments these days, but probably there were one fragment at some point as another distinct set. Those individuals from the Caatinga also show as a distinct set. So you see these outliers are all from small isolated populations. If you look at heterozygosity across the genome, most of the jaguars have fairly high heterozygosity. They don't seem to have problems in terms of genetic health, quite a few SNPs, millions of SNPs across the genome. But again, the outlier ones are those from these populations. So these three animals are from this population here. They have lower heterozygosity. And those two animals are from those two fragments there with considerably low heterozygosity. If you look at the inbreeding coefficient estimated by the fraction of the genome in runs of homozygosity, you see some animals with over 40% of the genome contained in runs of homozygosity, and this one is approaching 40% as well. So the different colors correspond to biomes, but those are all Atlantic forest ones from those same populations. So the red ones are here, the blue ones are there. So we're seeing a recurrent pattern of low diversity in those fragmented populations. So what are we doing with that? So of course, in addition to further studying those areas of the genome in runs of homozygosity, for example, looking at functional variants, loss of function variants, genetic load estimates. So we're interested scientifically in those aspects, of course. We're also doing more individuals from those fragmented populations. We're interested to see if the runs of homozygosity complement each other from different animals, especially here. This is back to that figure of the Atlantic forest. So we now have individuals from different fragments. So we want to see if they have runs of homozygosity at the same spots in the genome. And if they do, whether the same haplotype was fixed. Because I think it's quite possible that it was not. The different haplotypes were fixed, and therefore they could complement each other. So naturally, what we're trying to do is to reconnect them. So there are many ways of reconnecting them. None of them is easy. Basically, if you talk to a sensible person, they will say all of them are impossible, which is a problem. But we're trying not to be sensible and figure out a way to reconnect them. Either, well, directly connecting them through forest corridors is impossible, at least in the foreseeable future, because you have urban and rural development between these fragments. So translocations, artificial insemination, semi-captivity, where you actually take an individual and foster that they naturally reproduce in situ. These are all alternatives that we're talking about. I work with the Brazilian government in the genetic management of both in situ and ex situ population. Part of this was funded by the Brazilian Zoo Association that wants us to sequence the genomes of all the founders of the Brazilian ex situ population. So we're using these genomes to actually recommend pairing. So who should pair with who in the Brazilian zoo, so that we maximize genetic diversity, we maximize similarity to the original dynamics in nature, and we, of course, maximize representability of the different biomes. And another front is to design a SNP panel for forensic museum-based and non-invasive applications that will help in several different projects that are ongoing in Brazil. So that was quick, but I can go back to it if there's time during the questions. And now I have 6 minutes for the last example that I brought. This is the main study in Fernando Trindadi's PhD project. This is a collaboration with Russ Corbett Dietig at University of California, Santa Cruz. Beth's also involved in this project, several other colleagues. Bill Murphy is also a collaborator. So we have been studying the hybrid zone between these two species for a long time. I'm going to give you like a quick overview. And this project, Fernando's project, has to do with using low-cover genome of over 300 individuals to understand the history of this hybrid zone. So these are two small cats from that same Leopardus genus. This is a phylogeny we published last year. This was one of Fernando's PhD papers. This is a genome-wide SNP data set that we used to reconstruct the phylogeny of the genus. Now Jonas is repeating it with whole genomes in his paper. And we didn't have all the units, but that's the units we had at the time. So for example, that species complex that I mentioned includes Gutulus, the red one. I'm going to use color codes throughout these slides so that you can follow it more easily. It includes this, but it also includes the orange one, which we now have separated as a distinct species. And it also includes a Central American cat that we are now showing here. It's a third species. So this species complex includes 3, 4, perhaps 5 different species that we're still trying to define taxonomically. What we show here is that the red species, whose currently assessed distribution is here, and the blue one, which is from Patagonia and southern South America, they are not sister species. They've speciated about 2 million years ago. They subsequently speciated again, so they have their own sister species, and they are secondarily in contact here in southern Brazil, and that's where their hybrid zone is. We discovered this hybrid zone in the early 2000s, and we've been studying it with a variety of approaches, now going into this whole genome approach. So we have about 4,000 kilometer transects going through southeastern Brazil and even northeastern Brazil into Argentina. This is an outdated slide with 224 individuals with whole genome. Now it's about 320. So this is the pipeline that Fernando is using. So basically sequencing the genomes. We also have some higher coverage individuals, processing, filtering. We're looking at autosomal markers, X chromosome markers, Y chromosome markers, and mitochondrial markers at different depths. So our local ancestry estimates based on autosomal markers is performed with Ancestry HMM. It's a pipeline of software that Russ has developed. And we're comparing the different kinds of inferences that we have. So if you have an overall genome-wide assessment of ancestry, you have the red pure side, you have the blue pure side, and about 45% of the animals are hybrids. They are much more predominant on the blue side, so there seems to be a gradient that's skewed towards the blue. If you look at a PCA plot, you see the blue, you see the red, and you see the hybrids in between. If you look at it geographically, you find that most of them, so we're right here. This is where our city is. We're sitting at this hybrid zone, which is quite remarkable. And towards the south, you have blue. Towards the north, you have red. And here is where the hybrids are concentrated. If you look at mitochondrial analyses, you can easily tell them apart. You have hybrids. These are the two clades, the blue clade, the red clade. You have hybrids on both. If you look at the Y chromosome, which we could retrieve from this genome-wide data set, you also have hybrids on both sides. So we have hybrids with Y chromosome from blue and from red, which shows that the hybridization is bidirectional. Both sexes are involved in hybridization. They're backcrossing both directions. So it's a biparental admixture process. If you look at the local ancestry analysis, this is the whole genome with these ancestry informative markers across the genome, and you can tell whether a genomic block comes from blue or comes from red. And depending on the size of the block, you can date the admixture process. Because when it starts, you have, whoops, I also move my hands. So when it starts, you have a whole genome, an F1 genome that's fully red or fully blue, right, half of each. Then as they admix, these blocks are eroded by recombination, so you can use recombination-based inferences to define how old the hybridization process is. So we can do that as a mean or per individual, and overall, we're getting very recent estimates of the hybridization process, which is quite worrisome and quite remarkable, because we've been thinking about this for over 20 years, and now we finally have a tool using these genome-wide data sets to measure how old the hybridization process is. And there's a very important conservation implication for that. So we're getting, considering 2 to 3 years as a generation time, we're getting about 100 to 200 years age for this hybridization process, which matches habitat alteration after European immigration into this region. And the final piece of data I wanted to show comes from Caroline Sartor's work with landscape genetics and landscape ecology. This is the habitat suitability inference for the red. So it's an Atlantic forest species that's really mostly dense in this region, and for the blue, the southern species, and for the hybrids. So the hybrids seem to have higher habitat suitability in this region of southern Brazil where the hybrid zone is. And this matches, this is a map of habitat alteration of modified landscape. So this heat map indicates areas of higher conversion to agriculture, cattle ranching, forest culture as well. So these hybrids seem to have higher suitability in this region. And if you put that together, if you put the recent age and the habitat suitability results, we reach, unfortunately, the conclusion that this hybrid zone may be anthropogenic, or at least vastly exacerbated by anthropogenic processes. So this has very strong implications for conservation. So this was my final slide. I just wanted to leave you with this nice picture of one of those cats. This is the one in the red species. This is a Leopardus gutulus, leaping forward, right? So I was just thinking of how to end this talk on a somewhat positive note. We need to leap forward like this cat is doing. So these are worrisome times in terms of biodiversity conservation, and we've seen lots of examples here during the conference, but they're also exciting times from a scientific standpoint. Lots of interesting technological advances, lots of cool data sets, lots of interesting scientific problems to study. So I just put that here. Many threatened and poorly known species in areas like South America, and if you think of the global South in general, poorly known species. We don't even know their taxonomy. We don't know how many species there are, even in cats. We don't know how many species there are. We don't know what they eat. We don't know their ranges. We don't know much about their ecology, and a lot of them are threatened. Lots to do as quickly as possible, but there are amazing advances going on, many challenges and opportunities ahead. So I'd like to finish by thanking my collaborators in these studies, my students, postdocs, and former postdocs that have helped, funders, and institutional support, and thank you very much. I'm going to leave that one.
Sure.
I'll leave that one.
Yeah.
Oh, that one. Oh. All right.
Eduardo. Questions? Sure.
Yeah. Now I see the light.
Yeah.
Hi, great talk, really. I have a question about the Jaguar genome project, because I think you mentioned that you're considering reintroduction as a possibility, or I misunderstood, relocations. So are you considering reintroductions, so for example, like for the Asian elephant that Mirta presented on Wednesday?
Right. Okay, yes, as I said, there's plenty to talk about regarding jaguar conservation. We would like to consider reintroduction, and some few areas are potential areas for reintroduction, but unfortunately very few, because habitat loss is pervasive and human-wildlife conflict would be a problem as well. So we don't see it happening very soon. So of the over 50% of the range that was already lost, very little of it is adequate, or at least in the foreseeable future, for reintroductions. What we're mostly thinking about is just reconnection of fragments that are still there. So basically genetic augmentation, genetic rescue of fragments that are still there, especially in the Atlantic forest, which are somewhat distinct genetically. We want to preserve Atlantic forest alleles, Atlantic forest genetic constitution. and it's going away. So, and what we see is that the fragments don't have the same alleles. Because of the nature of drift, they are different representations of what used to exist in the Atlantic forest, and jointly they still harbor quite a bit of diversity. Actually, they're more diverse than the Pantanal, which is kind of interesting, but only if you look at the fragments combined. But individually, they are losing diversity quite fast. So reconnecting them is more of a priority than reintroducing in other areas. Camila, oh, there.
Yeah.
Thanks, Eduardo. That was a great talk. You said early on that you stopped using captive animals to do your reference genomes. Can you indicate why that was?
Can you repeat?
Why did you stop using captive animals for your genomes? I think it was the Ocelot you did it on.
Right. Well, sometimes for reference genomes we still use them, but if we want to use the same genome to infer, demographic patterns from the wild, then we see that the captive animals have had inbreeding in captivity. That's mostly why. So if you want to do like a PSMC reconstruction, in the case of Ocelot, it was fine, because now that we have a wild animal, it was very similar. But until we do, for example, in the case of the bush dog, we saw that our first bush dog was from a captive animal in the US, and it has a huge amount of runs of homozygosity. Then we did 3 animals from the wild, and it was not so. So I think it's mostly because of inbreeding induced in captivity that we can't really discern from real inbreeding in the wild.
So here.
Camila.
Thanks, Eduardo. So I wanted to talk about the taxonomic lack of resolution for many taxa, so the carnivores and many others in South America. So you have shown genomic evidences. I don't know if you have any other types of evidence. Do you think this is enough? Would this be accepted by the community to change the taxonomic status, or do you have a framework in mind on how to reach resolution or changes in the taxonomy? Because I think this can be game change, for certain areas, for certain taxa, if we manage to prove that there are many more species, or at least some more species than we think there are. So this can change the conservation approaches around many of these areas in taxa.
Yes, thank you for the question. I'm just going to go back to a slide. Yeah, I'll use this as an example, and I think it's a great question. It's a very important one for conservation. The orange here, this orange was the same species. It was, the whole complex was a single species, and using genetic data only, in 2013, we split the orange from the red. So now we're using red for this southern portion, and the rest was what was in orange, and we think it's going to split some more. But what we used was only genetic data. So it was mitochondrial DNA, microsatellites, X chromosome fragments, Y chromosome fragments, and we showed there was no gene flow between the red and the orange, and that was accepted by IUCN, that was accepted by the Brazilian Ministry of the Environment. They were assessed as separate species. They're now on IUCN as separate species. And yeah, and interestingly, the morphological colleagues had very slight evidence of differentiation that was not very convincing. So it was the genetic data that convinced the authorities that they were distinct species, and subsequently they collected more morphological data. Now they support it. So now there is morphological evidence for that split, but that occurred after we had shown it was different from a genetic standpoint. So I think it is feasible. We need to have robust genetic evidence. Of course, genomic evidence is even stronger, but you have to have multiple individuals from across the range. I think the otter example, we'll see, right? I think Klaus and I will see how convincing the otter example is, but it sounds convincing. I mean, it sounds like the two otter groups are non-monophyletic, and there's plenty of data there. And there has been some morphological data and ecological data as well. So of course, if you have ecological and morphological data, it's certainly better. It's easier to convince the authorities to reassess them as separate species, but it's an ongoing process for sure.
Very nice, thanks.
You're welcome. Couple there.
Claudia, and then Christian.
So I would like to ask you your opinion on mainly, like, as you have shown in your last project here, of low coverage sequencing, whole genome sequencing, and what is the role for conservation genomics? Because like on the one side, like for population genomic studies, low coverage is like fine and you can just see a lot of things, but maybe if you want to study runs of homozygosity and like genetic load that would have listen to that, that's not really an option because you don't have power to detect heterozygotes as well as if you have higher coverage. So what, like this is more of a general question, right? Like, what would be your opinion? Like, do you think that low coverage whole genome sequencing would be an option for conservation?
That's a good question. So this is our first experience with low coverage genomes, and we're only embarked in it because Russ is an expert on it, and he convinced me that it was worthwhile, and I think he was right. If you have, what we saw in this project is that it started to work when you had enough of a sample size. So you cannot do it with a few individuals. I don't think it will be good for the other applications that you mentioned, for runs of homozygosity, for detecting, for accurately genotyping each individual. So you don't. You use genotype likelihoods, and you use it across a large number of individuals. So for this kind of project, or for a population genetic project that's aiming at estimating demographic parameters broadly, I think it's quite powerful because, of course, it's cheaper and you can do hundreds of individuals for the price of 10 or 15 genomes. So it is very powerful for an admixture study like this one, for example, but I would not use it for more individual-based demographic reconstructions or load detection or runs of homozygosity or adaptive detection. So for all those, we do 10x or more, 10 to 30x generally for our genomes. But of course, there's a trade-off in cost. I mean, if cost goes down, we would do 10x for all those or more, right? But right now, it's a good trade-off for this type of project for which we have a large sample size.
Yeah, appreciate it. Yeah.
Thanks, Eduardo. You paint a very compelling picture about sort of the genetic signals that you're getting from all of these fragmented cats in these habitats. Have you tried to correlate it with anything other than just habitat fragmentation, other anthropogenic covariates like, I don't know, hunting pressure or proximity to road or human density?
Right.
Yep.
So my colleagues doing field ecology do that quite a bit. You have GPS-collared cats, and of course roads, there's road killing, but basically they're all correlated. So basically human matrix is impermeable to them. They just don't go across hundreds of kilometers of farmland, ranchland. They have a preference for large prey, so there's plenty of cattle ranching there. So the problem that happens when they try to disperse between the fragments that they come across cattle and they prey on cattle. There's no native prey left, so they prey on cattle and they get killed by the ranchers. So if you put together roads, cattle ranches, soybean farms, industrial areas, urban areas, it's a complex mosaic, but the bottom line is that it's impermeable to the Jaguar dispersal. So yeah, people are modeling those various layers, but they reach kind of the same conclusion in the east, in the Atlantic forest. In the Pantanal, where Carol actually is doing this, you have a more continuous matrix and the more continuous population of jaguars. They do disperse across the landscape. So it varies across regions. In the Amazon, it still seems to be a continuous population. In the Pantanal, it is. But in the Atlantic forest, it's totally different.
Thank you.
Sure.
Okay.
I think Graham, there's one more.
One more, okay. Graham, yeah, all right.
Thanks. That was a great talk and great to see how much brilliant research is going on in South America. So you seem to have a lot of small cats and encroaching urban areas. So I was wondering, in Great Britain, we have /had one species of small cat, which the Scottish wildcat, which is now totally integrated with domestic cats. Do you have that as a problem in South America? Do you have a lot of urban cats hybridizing with wild species? Is it a problem? And if it is, what can you do about it?
Okay, great. So far, we have not had a single case of domestic cats hybridizing with these wild cats naturally. In captivity, they do. There's some domestic breeds that have been created by humans from hybrids between some of these cats and domestic cats. But in the wild or in urban areas or rural areas, we have not had a single hybrid. So they are about 8 million years apart from domestic cats, and they seem to be different enough that they don't interbreed. So that's reassuring, at least. That problem we haven't had yet. With dogs, there has been a recent case of a fox, of a crab-eating fox that looks weird. and they have suspected that it's a hybrid with a dog. And some people, I haven't seen the data yet, but some people are telling me that they've typed some markers, and it seems to be a hybrid between crab-eating fox and dog. And this is a South American canid that's 8 million years apart, 10 million years apart from dogs. So, but it's not common at all. And in the cats, it's absent, as far as we know.
So is that a one-off cross, or is that like a population which is?
Yeah, this is one individual from the fox case and 0 in the cats. So we have other problems, but not hybridization with domestic cats, fortunately.
Okay. All right. That'll be
Thank you,
for our
Klaus.
questions. Thanks for these excellent
you.
questions and for a great talk.